So now we're going to take a look at the central dogma of biology, which is that genetic information goes from DNA to RNA in a process called transcription. And using the message in the RNA, we can build proteins through a process called translation. So we've been talking about pretty much all year that the job of DNA really is to hold information about how to make proteins. The genes are the instructions for how to make proteins. So now we're going to learn physically how does that happen? Well, the gene has a code, and that code is going to get turned into an RNA version. It's the same message, but it's like turning it into a different language. So it's originally written in a DNA language. We're going to write the exact same message into an RNA language, and that process is called transcription. That RNA molecule that we make is going to go to the ribosome, and the ribosome is going to read the RNA message and use that message to build a polypeptide, which is the starting material for building a protein. The sequence of amino acids is determined by this RNA code. So there's been a few different hypotheses in the past. There was one hypothesis that was called the one gene, one protein hypothesis, which said that for every one gene, we can build one protein. But this is actually incorrect, and I think you might know why this is incorrect. Remember that some proteins are actually made from more than one polypeptide. So there are proteins that are made from the quaternary proteins. They're made from many polypeptides, at least two or more put together. So therefore, one gene does not always equal one protein. However, the new hypothesis is the one gene, one polypeptide hypothesis. For every one gene, we make one polypeptide. Let's take a look at this DNA inside the nucleus. Now, after transcription, we make this RNA molecule here, which is carrying the exact same message as the gene. And then that RNA molecule is going to leave the nucleus. It's going to go into the cytoplasm. You'll find a ribosome, and then we will take that code and turn it into a polypeptide string. So how does DNA, which only has 4 different nucleotides, how does it actually store this information? Well, the order of the nucleotides is actually a type of code, just like computer code. And this code is made of 3 nucleotide sequence pieces called codons. So basically, the order of the nucleotides is the information. Okay, this is the information. It's a codon DNA language. However, when we think about ribosomes, they don't really understand the DNA language. So ribosomes don't actually directly interact with the DNA. Instead, this DNA message needs to get converted into an RNA message first. During translation, the RNA message is going to be translated into an amino acid sequence. So the code, it's not just one nucleotide at a time, but rather it's 3 nucleotides. This is called the triplet code. So in the DNA and also in the RNA, we have these 3-letter words. You can kind of think of them like a word in English. It's 3 nucleotides at a time, and they're called codons. So let's take a look at some of these codons in the next video.